Hey everyone, and welcome to today's Explainer. Have you ever wondered exactly how we know what active ingredients are actually hiding inside complex herbal medicines? Well, today, we're diving deep into one of the most vital and honestly coolest analytical techniques in pharmacognosy that makes this all possible. We're going to break down how incredibly complex mixtures from nature are separated with absolute pinpoint precision. So, if you're curious about the exact science behind the purity of plant extracts, you are definitely in the right place. Here's a quick look at our agenda for today. We'll kick things off with an intro to chromatography, then look at TLC with a baseline diagram. After that, we'll explore the Hptlc upgrade, the principle of Hptlc, and wrap up with Hptlc components and working. Let's get right into it. Section 1. Intro to Chromatography. Okay, so before we can analyze anything complex, we obviously need a reliable way to pull its individual pieces apart, right? Imagine you've got this incredibly complex, messy soup of chemicals extracted from a plant leaf. You can't just eyeball that mixture and know what's in it. Chromatography is the laboratory technique that allows us to elegantly and precisely separate it into its individual, isolated components so we can actually study them. And this is exactly why this matters so much in pharmacognosy. It's essentially our primary tool for extracting things like polyphenols, isolating a specific medicinal compound from a natural source like fungi, or identifying unknown substances. Chromatography is literally the starting line for all of it. Moving on to Section 2, TLC, the baseline diagram. Let's look at this classic baseline diagram. The standard setup is just beautifully simple. You've got a glass developing chamber, and standing up inside it is a TLC plate. That's our stationary phase, typically coated with something like silica gel on an aluminum backing. Down at the very bottom, there's a shallow pool of solvent, which acts as our mobile phase. As the solvent creeps up the plate via capillary action, it drags the components of your spotted sample along, separating them out as they go. Once it's developed, you just dry it and visualize the newly separated spots. But yeah, here is the catch. While classic TLC is wonderfully simple, super low cost and versatile, it eventually hits a wall. When you're just looking at those little spots with a human eye, or even under basic UV light, you run into two massive limitations: less quantification and less sensitivity. Sure, you can see that a compound is there, but measuring exactly how much is there, that's incredibly tough. And if you only have a microscopic trace amount of a substance, standard TLC might just miss it entirely. Which brings us to Section 3, the Hptlc upgrade. I always say, think of regular TLC as your basic rough pencil sketch. And Hptlc, that is the fully automated 4K digital remaster. Hptlc is an advanced form of traditional TLC. It takes that exact same basic idea of a plate and a solvent, but makes it highly efficient, highly automated, and vastly more reproducible. This means if you run the exact same test 10 times, you get the exact same perfectly quantifiable results 10 times. And that is absolutely essential for analyzing highly complex mixtures. Because of this massive upgrade, it's become an absolute powerhouse technique. We aren't just talking about small academic labs here. It's the heavy lifter standardizing the exact purity of pharmaceuticals and ensuring the safety of the foods and cosmetics that we use every single day.
All right, Section 4, the principle of Hptlc.
Now, it's kind of fascinating that even with all the high-tech automation we are about to look at, the core principle is still based entirely on differential affinity. It's basically a tiny chemical tug-of-war. The different components hidden inside the sample have a choice to make. Do they prefer to stick to the stationary plate, or do they prefer to ride along with the mobile solvent? If a compound has a higher affinity for the moving liquid, it travels further up. If it prefers the stationary plate, it stays lower down. That simple difference in attraction is what physically pulls the mixture apart into distinct, measurable bands. So, by taking that basic principle of differential affinity and applying rigorous, standardized instrumentation to it, Hptlc achieves its signature high-performance characteristics. First, you get high-resolution separation, meaning you can clearly distinguish multiple components, even in wildly complex mixtures. Second, improved sensitivity means you can detect much, much smaller amounts of a substance than traditional TLC could ever hope to see. And finally, increased accuracy. Because the method is completely machine standardized, the exactness of the results just skyrockets. Let's see how this all comes together in Section 5, Hptlc components and working. When you look at the proper working diagram of these components as a chronological workflow, you can see exactly how Hptlc automates every single manual step. Instead of a human hand shakily pipetting a spot, Step 1 uses a machine to spray the sample on in perfectly uniform microscopic bands. In Step 2, the automatic development chamber meticulously controls the exact humidity, temperature, and solvent saturation. After drying in Step 3, we hit the critical piece, Step 4. A densitometer uses light to optically scan the bands, giving us exact, quantifiable data before Step 5 documents it all digitally. The whole process is incredibly precise,
Ultimately, taking human hands completely out of the equation, swapping them for an automatic applicator, and reading the results with a precision densitometer rather than the naked eye is literally what transforms a basic lab experiment into a high-powered analytical method. It totally removes human error. It guarantees that the volume of the sample applied, the specific conditions of the development chamber, and the final measurements are flawlessly 100% standardized every single time.
So as we wrap up today's explainer, I want you to think beyond the laboratory bench for just a second. When you rely on a standardized herbal supplement or a precisely formulated cosmetic product for your health, it is exactly this invisible, automated precision working behind the scenes. If HPTLC is so essential for analyzing these complex mixtures, how might this incredible high-precision automation be quietly ensuring the safety and quality of the products sitting right there in your bathroom cabinet today? It's pretty wild to think about. Thanks so much for joining me, and I'll catch you in the next one.